Okay, my dear friends, welcome to today's session. Today's eminent resource person, Dr. M. Rambabu Garu, Assistant Professor, Department of Botany, Kakatiya Government Degree College, Autonomous, Warangal. Sir is going to explain about plant tissue culture and biodiversity. Sorry, plant tissue culture and biotechnology. So this is one of the most important area, my dear friends. Listen carefully. During our class, if you have any doubt, please ask our respected uh, resource person. Okay, sir. Please start your session, sir.
So thank you, sir. Thank you. So plant tissue culture. Yesterday we have started this topic. Sir, is it visible, sir?
Yes, sir. Yes, sir. It is visible, sir. Please proceed.
Right. Right. So plant tissue culture. Yesterday we have started some introduction part. So plant tissue culture is a culture of the plant parts like seeds, organs, tissues, etc. Unsuitable nutrient media, always under sterile condition. So in this process, we will culture the plant parts like stem, leaves, root, and tissues, etc. on suitable nutrient media. This is called as plant tissue culture. This experiment is always under. So coming to the background. So what is the background of plant tissue culture? Tissue culture had its, he is the great scientist Gottlieb Haberland. So he is the father of plant tissue culture from German, Gottlieb Haberland. This tissue culture technique is started from 20th century onwards. Father of tissue culture, Gottlieb Haberland, he cultured the cork tissue. He cultured the cork tissue on nutrient media and successfully established the tissue culture process. And he regenerated the complete plantlets from the culturing of the cork tissue, unsuitable nutrient media. So that's why he is considered as the father of tissue culture. So these are some historical backgrounds.
Sir, my voice break అవుతుంది సార్ రావట్లేదు.
Rambabu, sir. Rambabu, sir.
Sir, one disturbance.
Okay, sir. Please proceed.
So some important historical background of the plant tissue culture, very, very important. You see the last one, broadcast isolation, caulking scientist. So this question has come in our degree called lecturer entrance examination. First, enzymatic isolation by protoplast established by caulking. So these are some important background, historical backgrounds of the plant tissue culture. We must be find out these. I will send this PowerPoint presentation to your group. So MS Media, Murshid and schools like this. Then coming to the basis of plant tissue culture, what are the bases? Majorly, two plant growth regulators are affected on plant tissue culture, plant differentiation process. They are the auxins and cytokinins. So auxins stimulated root development, cytokinins stimulates shoot development. Here, these are two important plant growth regulators. Auxins and cytokinins both are plays a vital role for the purpose of proliferation, callus formation, regeneration, root induction. Like this, organogenesis process, for the purpose of organogenesis, these two plant growth regulators like auxins and cytokinins is very, very important. These ratios, auxins and cytokinins ratios, shows various type of effects. You see, high auxin, low cytokinin, high percentage of auxin and low cytokinin. These concentrations result that root development. Auxins are used for the root development. High cytokinin, low auxin. Concentrations result that shoot development. Cytokinins generally used for the shoot development. Auxins and cytokinins are equal concentrations, result that callus development. This is the formula. High auxin stimulates the root development. High cytokinin level stimulates the shoot development. And auxin and cytokinins equal concentrations stimulates the callus signature. This is the formula. Then coming to the what are the needs for the establishment of the plantlets through tissue culture? They are appropriate tissue. Some tissues are better than others. For the purpose of plant tissue culture, we must be take the healthy and young tissues. From the healthy and young leaves, for the healthy and young shoot birds, we must be take that fresh and healthy tissue for the purpose of tissue culture. So that is very, very important. Selection of the tissue is very important. So if the tissue is not in good condition, our experiment will not carried out. So for the purpose of the tissue culture, we must be take healthy and appropriate tissue is important. Next one, suitable growth media. Media is a food material like component. It provides all type of essential elements to the inoculated tissue. That's why we must be take suitable growth media. So that media includes micronutrients, macronutrients, vitamins, amino acids, enzymes, whatever that tissue wants, all type of elements are present in that nutrient media. So that media is also plays a vital role. And third one, aseptic condition. Tissues are inoculated on suitable media. If that media will be contaminated, the total experiment will be failure. That's why this tissue culture process is always under aseptic condition. Always under aseptic condition. So this is it. Then fourth one, growth regulators. With the nutrient media, we must be add some growth regulators like auxins, cytokinins, gibberellin, etc. Auxins means indolestic acid, IA, IPA, NAA, 2,4-D. Like these auxins are very, very important in the cytokinins. Kinetin, zeatin, like cytokinins also very, very important. Gibberellic acid. So these plant growth regulators are stimulates the growing process into the tissue on the nutrient media. Then environment factors. So we must be create a normal environment-like condition in the laboratory, like temperature, light, photo period of the light intensity, time of the light, temperature. Some of the measured qualitative and quantitative environment factors must be maintained in the tissue culture room. Then frequent subculture. After some days, the tissues will be transferred to the another nutrient media. So that is the frequent subculture. Transfer after 3 weeks, transfer after 3 weeks, transfer. So these are the basic needs for the purpose of plant tissue culture. Then coming to the steps. What are the steps involved in plant tissue culture? Nutrient media preparation, sterilization of nutrient media, explant preparation, inoculation, incubation, hardening, and acclimatization. These six are the basic steps for the purpose of the production of new plant through tissues. We will see one by one. So nutrient media preparation, take double sterilized distilled water and add suitable quantity of the following substances. Take the distilled water and add the following substances. The first substances is inorganic nutrient. So for the purpose of inorganic nutrients, so those are macronutrients and micronutrients like components are present. Here organic nutrients. In that carbohydrate like sugar, vitamins, thiamine, then amino acids, glycine. So all these are organic nutrients. Then plant growth regulators, already I discussed, auxins, gibberellin, cytokines, abscisic acid. So like this, not abscisic acid, only auxins, gibberellins, and cytokines. So these are very, very important for the purpose of growth. Finally, agar-agar. So agar-agar is a powder-like substance. This is used for the media converted into semi-solidification. The liquid media is converted into semi-solidified media by the adding of this agar-agar. Please mute your voice. Hello. So agar-agar is of course, this is a powder-like substance. If we will add this agar to the liquid media, that will be automatically converted to semi-solidified media. So for the purpose of semi-solidification, this agar-agar must be added. Then the pH must be measured in between 5.6 to 5.8. The pH level is also very, very important. If the media is acidic or basic condition, that will not be considered. Then coming to the sterilization of nutrient media. After the preparing of these components into the distilled water, after the preparation of that liquid media, then that will be goes to sterilization process. For the purpose of sterilization, we will take the help of autoclave equipment. So autoclave equipment is generally worked on pressure cooker mechanism, like same pressure cooker. I will show the diagram. So take that prepared nutrient media into culture tubes and go to the autoclave. The autoclave, in that autoclave, the media will be sterilized at 121 degrees centigrade temperature and 15 pounds of pressure. The time period is 15 minutes. This is a measured timings. 121 degrees centigrade temperature, 15 pounds of pressure, and 15 minutes of time. In that autoclave, so the entire media will be sterilized. So this is the autoclave equipment already in. Then coming to the explant preparation. So explant means, so which type of tissue or which type of parts are going to cultured on nutrient media is called as explant. The cultured plant material is called as a explant. So for the purpose of culture, this explant must be sterilized. Then first of all, you take that plant parts, leaves or nodal parts, you take that plant parts and wash with running tap water, then wash with liquid detergent, then wash with the fungicide. Fungicide means wash the explant with the fungicide, like mercury chloride and sodium hypochlorite solution. So with the washing of these fungicides, that explant or sterilized. Finally, wash with the sterile distilled water, and those explants, those plant pots are cut into small pieces. First of all, take the leaves. For example, take the leaves and of fungicide. Then finally wash with sterile distilled water. Then finally cut into small pieces. The cutted small piece is called as explant. The fungicide, then cut into small pieces. That small pieces are called as explant. The size of the explant is in between 3.3 to 5 millimeter size. From inoculation chambers. That inoculation chambers are like laminar airflows. You see the diagrammatic presentation. Now, in that inoculation chamber, the chamber is always maintain the aseptic condition with the help of UV rays, with the help of spirit. The chamber always maintained aseptic condition. In that inoculation chamber, we will transfer that sterile explant onto the sterile nutrient media. This is called as inoculation. For the purpose of inoculation, we are taking the help from the inoculation chambers. Then incubation. After the placing of this explant on nutrient media, those will be transferred the separate inoculation chambers. You see here, these are the inoculation chambers kept into a suitable chambers like library. Books will be stored on the library like these inoculated test tubes or culture tubes or culture bottles will be shifted on the specialized culture rooms called as inoculation chambers. So in that culture room, the temperature always maintained in between 25 plus or minus 2 degrees centigrade. These are the measured conditions. The temperature is always maintained in between 25 plus or minus 2 degrees centigrade. The related humidity, humidity means the water content in the air. The humidity in between 50 to 60%. The light intensity is 3000 to 5000 approximately light intensity. Then the time of light is 16 hours of the photoperiod. These are some measured suitable quantitative measurements. Temperature 25 plus or minus 2 degrees centigrade. The humidity 50 to 60%. The photoperiodism time of the light intensity is 16 hours. So these much of the measurement items will be maintained in these culture rooms. So after the inoculation or incubation, after the inoculation and incubation, the tissues will be regenerated by the taking of the elements, by the taking of the nutrients from the nutrient media. That inoculated explant will be regenerated. After 1 or 2 weeks, the explant is converted into undifferentiated mass of tissue. After the inoculation, after the transfer of that inoculated into culture rooms, the tissue will be converted. After 1 or 2 weeks, the tissue will be converted into undifferentiated mass of tissue. That is called as callus. The formation of callus is called as callusing. By the effect of auxins, by the effect of the nutrients, this tissue will be converted into callus. These callus further transferred on, I will show this diagram. You see, this is the diagram. So these are the explant left side cultured on the nutrient media, right? That is called as inoculation. So after the observing of these different types of the auxins, IAA, IBA, NAA, so this explant is converted into callus. That is called as callusing. Then this callus will be subcultured, again culture shifted to the another culture bottle. That's called as subculture. So by the adding of cytokinins, like BAP or kinetin, so that callus will be regenerated into plantlets. That callus subcultured on the cytokinins media. Here first is inoculation. The second one is callus formation. The third tube is regeneration. This callus will be subcultured on cytokinins media. By the effect of the cytokinins, this callus produces the small plantlets. That's called as regeneration. These regenerated plantlets will be shifted again on the rooting media. The fourth tube, you see, the plantlets will be shifted again onto the rooting media. What happens automatically? The roots will be developed. That's called as rooting. So for the purpose of callusing, auxins are helpful. For the purpose of regeneration, means plantlet formation, cytokinins are helpful. Then the purpose of the rooting, auxins are here useful. After this complete formation of these new plantlet in the test tube, that is gradually transferred to the outer environment. That is called as lab to land program. Yesterday I have told that. These test tube plants gradually shifted to the outer environment. So that is called as hardening or acclimatization. So for the purpose of this hardening and acclimatization, we are taking the help from the greenhouses. These test tube plant gradually picked up from the test tube, wash those callus from the roots and shifted to the sterile soil, right? Here, the pot-like structure here, sterile soil, right? And cover the polythene bag and put in the separate, greenhouses. Gradually, that plant hardened means it will be take the resistance power to survive the outer environment field. Immediately that plant picked and put the outer environment. If suppose immediately the test tube plant pick up and put the outer environment, that will be dead. Why? Because the test tube plant is surviving by the outer glass of the tube. This is very sensitive. And already I told that test tube plant, there is no cuticle layer. But outer plants shows the cuticle layer. That's why they are very happily survive. So this test tube plant gradually develop the cuticle layer around the outer surface of the epidermis. Gradually develops the cuticle layer around the epidermis. That's why these are gradually shifted from the test tube, from the laboratory, then from the greenhouses or glass houses, then gradually shifted to the field. This entire process is called as hardening and acclimatization. So this is the plant tissue culture technique. Here, a tissue culture means the inoculation or culture of plant material, parts, or tissues, unsuitable nutrient media, always under aseptic condition, is called a plant tissue culture. Generally, this process is based on cellular totipotency. I have told you, cellular totipotency means each and every cell have the capacity to produce the new plant that is similar to its parent plant. By the repeated cell division, cell elongation, and cell differentiation, these tissues further regenerated into complete plants. So who is the father of plant tissue culture? He is Haberland. Gottlieb Haberland is the father of plant tissue culture. What are the basic plant growth regulators are useful for the plant tissue culture? Auxins.
Auxins is cytokinins.
Cytokinins. Auxins are used for the rooting and callousing. Cytokinins are used for the regeneration and plantlet formation. So this is the process of plant tissue culture. Any doubts, my dear students? Do you want asking anything? Any doubts?
No, sir.
Okay, sir. Thank you very much.
That's right. Okay, sir. Thank you.
Rambabu, sir. Thank you. Thank you very much. Today's class is very excellent. My dear students, this is one of the wonderful class.
If you have any doubt, please ask our respected resource person. Tomorrow, same time, our sir is going to start the session.